Right, so first thing to talk about is Microbe of the Week. Thanks everybody who sent answers in. I try to give a reply, and until it's time for me to like watch some television or eat my dinner, I try and give answers immediately. Okay, so probably the first pretty correct answer came from Lisa. Other people gave good answers a bit later on. Yeah. Yeah, sure. Oh, actually, no, because I'm going to tell you the answer right now. So that's the kind of format, so that, yeah, just don't listen. Okay, so, still plenty to play for. There's one person who got like 0.25 points for their answer. It's kind of like really minimalist answer here. Okay, so that's where we are on the scores. What? Yeah, what surprise. Well, yeah, you've discovered that in the last lecture, right? Okay, so that's how what the standings are at the moment, okay? So what was the question? Now, okay, so if, just to remind everybody about the question I sent out, the question was, okay, mycobacteria, they have peptidoglycan in their cell wall, but they are not sensitive to vancomycin or beta-lactam antibiotics, which both inhibit cell wall synthesis, peptidoglycan synthesis. So if you think, let's just think about the simplest case here, okay? Gram-positive cell wall, the peptidoglycan layer is quite permeable. So beta-lactams and vancomycin can easily just diffuse across the peptidoglycan layer and come down and inhibit the enzymes here that are involved, okay? So they all inhibit transpeptidases. So for gram-positives are sensitive to both these types of antibiotics, vancomycin and beta-lactams. Gram-negatives, they've got the outer cell membrane. Vancomycin is quite a large molecule and it can't diffuse across the outer membrane and it's too big to go through the porins. However, many beta-lactam antibiotics are small, well, they're all small molecules and mostly they can go through the porins of gram-negative bacteria, so they can get access to the periplasm and inhibit the enzymes that are involved in peptidoglycan synthesis. Now, to understand why that doesn't work with mycobacteria, you have to go and look at the structure of the mycobacterial cell wall, which, as I said a couple of days ago, is very different than the cell wall structure in gram-positives and gram-negatives. Now, I don't know this stuff off by heart, so I don't expect you to either, so you can just kind of check out by Google mycobacterial cell wall structure, and you might find out something like this. So this is from Nature magazine, so it's a good source of information. And you'll have this kind of diagram for the cell wall structure of a mycobacterial cell wall. So this is the plasma membrane, this is the periplasm, this is the peptidoglycan layer. And then you'll see that, in fact, mycobacteria have got an outer membrane, but it's constituted of these mycolic acid, kind of very long chain fatty acid lipids. So it's not like a regular lipid bilayer here. It's very waxy, very thick, and highly impermeable. Now, mycobacteria also have membrane porins in this mycobacterial membrane. But the structure of these porins is entirely different from the structure of the porins in the gram-negative outer membrane. So they are not permeable to the same molecules, okay? So in fact, this is the reason why mycobacteria are not sensitive to vancomycin or beta-lactams, because The outer membrane is basically too thick and waxy and is impermeable to a lot of molecules, and the membrane porins are very, very different and actually don't let beta-lactams pass through. Okay, so that was the correct answer. Okay. Right, so if I remember rightly, we had covered these two slides, but we hadn't looked at the details of peptidoglycan synthesis. That's right, huh? So I'm going to go back a couple. Oh, no, I'm not going to go back. Yeah, sure. Go back, go back, go back. Oh, it's all happening backwards. Okay, so this is the general process, okay? So I'm just going to remind you a little bit about this. I talked about this, right? No? Okay, thank you. Okay, so these are the 4 steps involved here. Now, it's complicated, and whenever you have something complicated in biology, you've got to try and think, why is it like this? Why isn't it just simpler and easier, and it'd be much easier to learn and understand this stuff? Well, often things are done in a complex way in biology because they're solving a difficult problem. And the problem here for bacteria is that they need to synthesize a polymer on the outside of the cell. So in metabolism, you need to put energy in to synthesize macromolecules. But outside the cell, there's no ATP or biochemical energy. That's the problem. So how does the bacteria get around this? Well, they get around it by synthesizing the precursors, the repeating subunits in the cytoplasm. So the first step is to synthesize, the NAM-NAG repeating glycan part, and then the pentapeptide in the cytoplasm. Now, if you remember, in the mature peptidoglycan molecule, it's a tetrapeptide. There's 4 amino acids attached to each N-acetyl-muramic acid glycan, okay? But it's synthesized first as a pentapeptide. So you need to try and understand why this happens, okay? Wouldn't it be simpler if it just produced a tetrapeptide, right? Why is it doing something complicated like this? Okay, so the repeating subunit synthesized in the cytosol. Then it's going to be transported across the across the plasma membrane into the periplasm by bactoprenol, which is a kind of lipid transporter. So it's flipped over, gets to the periplasm. Then once in the periplasm, the NAM-NAG glycan part will be inserted into the NAM-NAG polymer by transglycosidases, and then finally the peptide crosslinks will be formed by transpeptidases. And the transpeptidases are the site of action of beta-lactam antibiotics. They're competitive inhibitors of the transpeptidases. Vancomycin too, but by not competitive inhibitors. They inhibit that step, but by a different mechanism. Okay, so there used to be a good YouTube video about this on a site or a channel by somebody called Microbio, but it doesn't exist anymore. This one is still up there. I think it's a little bit less good, but it's okay. And okay, so beta-lactam antibiotics, like penicillin, they're inhibiting pretty much the last step in the synthesis here. But they're still bactericidal. So why is that? One thing you have to realize is that when bacteria are growing, in order for the bacterial cell to get bigger before it divides into 2, bacteria have got to lyse the peptidoglycan layer at the same time as they're synthesizing it, because otherwise this structure is just so solid that the cell can't even expand, right? So if the cell is able to grow, it's got to be able to cut the peptidoglycan so that it can push it apart as the cell grows bigger. And that's the role of autolysins that bacteria express in the periplasm, and they break up the peptidoglycan. So when bacteria are growing, that's okay because, you know, the peptidoglycan is being cut up a little bit, but then it's also being synthesized. So the net effect is you make more peptidoglycan. But if you inhibit the synthesis step, all you've got left is the activity of the autolysins, and then that's why these bacteria will explode. They'll be killed by the action of these antibiotics. Okay, penicillin, cephalosporins, vancomycin. Okay. Right, so that's a little bit about that. Now, let's have a look at the different steps in the synthesis in more detail. So, it starts off by synthesizing, bacteria start off by synthesizing N-acetylmuramic acid coupled to uridine diphosphate in the cytoplasm, and the 5 amino acids of the pentapeptide get attached to this lactoyl group, which is at position 3 of the N-acetylmuramic acid sugar. So never forget, okay, the peptide part is attached to the NAM, okay, N-acetylmuramic acid, not to the NAG. How can you remember that? The important point is acid. The acid comes from the carboxylate group here on the lactate. So this carboxylate group can be linked to the N-terminus of this amino acid, right? That will make a peptide bond here. Everybody okay with what a peptide bond is? So once you've got the first one, you have peptide bonds here from the, you know, C-terminus to N-terminus, C-terminus to N-terminus all the way around. So N-terminal here, C-terminal down there, okay? Right. Next step is that this whole thing is going to be connected to bactoprenol. So this is the lipid part. Initially, it's monophosphorylated with the phosphate group here sticking out into the cytoplasm. So after this step, you get bactoprenol, pyrophosphate, and then NAM and the pentapeptide. So where does this other phosphate group come from? Yeah. Yeah, from the UDP, uridine diphosphate. So you've got like phosphate, phosphate, uridine. So you have like bactoprenol, pyrophosphate, NAM, and what do you get out of here? As you know, once this comes off, what's the other product of this? Yeah, uridine monophosphate, UMP. Okay. Yes. Okay, you could imagine you have like another arrow coming down here, like a curved arrow, and then UMP will be, you know, released into the cytosol. So that's where this second phosphate group comes from. Okay, so this is also called lipid 1 in the synthesis of peptidoglycan. Then the next thing that happens is that you get N-acetylglucosamine stuck on the other end of the NAM molecule here. So that's it. This is our repeated subunit of peptidoglycan, NAG, NAM, pentapeptide, all attached to bactoprenol diphosphate or pyrophosphate. So that's good. That's step 1. Next thing that happens, this is also called lipid 2 in the biosynthetic pathway. So the next thing that happens is lipid 2 is going to be grabbed by a kind of enzyme called it has a flippase activity. So it's going to flip the whole thing in the plasma membrane. So, you know, it's inverted, it ends up like this. So the lipid part of the bactoprenol just stays in the membrane, but the NAG-NAM pentapeptide is now in the periplasm. So bactoprenol is often called, you know, the lipid transporter of, you know, peptidoglycan subunits. Okay, last 2 steps are the transglycosidation. So transglycosidases are going to attach this NAM-NAG dimer to one of the free ends of the NAG-NAM polymer. Okay, so they create a glycosidic bond, and at the same time, they destroy the bond from this phosphate group to the sugar. Okay, so you're destroying one bond, creating a new bond, but that's energetically equal, right? It's equal. It's balanced. So it doesn't cost any extra energy to do this. Okay, remember, this bond here, the phosphate, originally came from the UDP. Okay, so spending the energy in this bond occurred before in the cytoplasm, because you had UDP to UMP. And that's how you can export the energy content out into the periplasm here, where you don't have any UDP, ATP, nothing, all right? So that's the trianglycosidase action. What happens afterwards is that the bactoprenyl pyrophosphate is reflipped in the membrane, and this regenerates bactoprenyl phosphate to be used in another round of the synthesis. Okay? And the last step is the action of transpeptidases, which are going to form a peptide bond between the free amine group on the side chain, either of the DAP, diaminopimelic acid, or the L-lysine, and that's going to form a peptide bond with the carboxy terminus of the fourth D-alanine here. So this is going to be catalyzed by a transpeptidase. So what's going to happen is that in the pentapeptide, you have this peptide bond between the fourth and the fifth D-alanine. That's going to break and be replaced by a peptide bond here. So you get this. And you release one D-alanine molecule into the periplasm that will have to be re-imported into the cytosol. So this reaction also doesn't cost any energy, because you form one peptide bond and you break one peptide bond. And that's why bacteria have got to synthesize the precursor as NAG, NAM, and a pentapeptide. Because the fifth amino acid, that's what's carrying the energy you need to make this bond, okay? That's why the thing is so complicated, because you've got to have some kind of energy within the kind of lipid 2 that can allow the creation of these two types of bonds. And that's why it's so great, it's so clever, I love it. Okay, so that should be all right for everybody. Right, so let's have another clicker question here. I'll just start this one for you so everybody can think about that. See if you've understood everything. Okay. Ooh. Yeah. It's not working? Oops. Oh, it's all gone. Hang on, what could possibly be going wrong here? Maybe it's the wrong code. I'll stop. I need to stop there. I don't know, is that the same one or a different one? Let's check it out. Try that one. Might be the same, might not be the same, but that's the one that I have on this question. Oh no. It's really not working. Okay, well we broke the gadget. Okay, so you can all think about this, right? And we'll just do it by a show of hands if it's not working. Okay, so there's more than one correct answer here, okay? All right. Why that didn't work? Oh no, I didn't start. Sorry. It will work. I didn't press start. Oh yeah. Yeah. Yeah, it's been a long day already, you know. Okay, that's pretty good. I got 34 answers out of like 30. Okay, so here's the 35. Okay, yeah. So, oh, everybody, right, great. So here's the answers. Okay, so we'll start with this one. Number 5, evolve a way to survive without a cell wall. Yeah, this is what mycoplasma do, but this is not really available for most bacteria. They can do this because they're parasitic, they live on eukaryotic cells, they can get cholesterol. Most other bacteria cannot do this. Okay, so this is basically no, not really feasible. Then what else do we have here? Okay, now going up to the top, and express an enzyme that destroys the antibiotic. Yes, that's true. That's a known resistance mechanism. Beta-lactamase, when you work in molecular biology with resistance plasmids for ampicillin-resistant bacteria, then the plasmid expresses a beta-lactamase, cuts the antibiotic up. Express a transpeptidase that does not bind the antibiotic. Yes, that's also a known resistance mechanism. Definitely in gram-positives, don't know about gram-negatives, probably. 3, express a transglycosidase that does not bind the antibiotic. That's wrong, okay? Beta-lactamine antibiotics inhibit the transpeptidase. They bind to the active site, they inhibit the enzyme. They don't inhibit the transglycosidase. Okay, so this number 3 is wrong. It's the wrong enzyme activity. This is the one. And number 4, express a porin that excludes the antibiotic from the periplasm. Oh, I'm so glad. Oh, let's have a look at the numbers here. How many people? 9 people. Oh, 9 people. Ooh. Okay, you fell into the trap. What's wrong here? What have you done wrong? Okay, so why is that wrong? Porins, they're in the gram-negative cell wall. In the question, we're talking about gram-positive bacteria. Gram-positive bacteria, they don't have porins. So this cannot be a correct answer for gram-positive. But it is a resistance mechanism, but in gram-negatives only, okay? So there we go, that's peptidoglycan. Okay, so let's get on to the next external structure. And this is, yes? Okay, these transpeptidases do not need any extra energy source because they cleave one peptide bond, that's this one, and that cleavage releases the energy you need to make this bond. Okay. Also, no extra energy input because you're cleaving this phosphate phosphodiester bond here, and so that's cleaved, that releases the energy you need to make the bond with this sugar over here. Okay, so there's no ATP, UMP, anything, UDP, nothing needs to be there. Okay, so they're both energy neutral, these steps, and they have to be. Okay, they have to be neutral in energy terms. Otherwise, it can't happen outside the cell. Okay, lipopolysaccharide, this is the next external structure that we need to think about. It's an essential component of the outer membrane of all gram-negative bacteria. So, as I may have mentioned before, the outer membrane of a gram-negative bacteria isn't composed of ordinary phospholipid. The outer leaflet is composed of lipopolysaccharide. So what is that? Well, there's a lipid part at the base, and this is attached to, firstly, a core oligosaccharide. So this core oligosaccharide structure has got some weird sugars in it, like this one, KDO, and then some more familiar ones. So the structure of the core oligosaccharide is pretty well conserved. Certainly, all the species within the same genus will have the same structure of the core oligosaccharide. And then after the core oligosaccharide, there are like, I don't know, 5 to 30 repeats of, repeated sugar motif. So in this motif here, you could have like maybe 5 to 8 different sugar molecules, either in a linear or a slightly branched structure. And these O antigen repeats are variable between bacterial strains, between species, and also within a species you can have a lot of variability. Okay. So what is this for? Well, the lipopolysaccharide layer is really there to protect the outer membrane from chemicals that can attack it. So we're talking about like detergents, bile salts in the digestive system. Also, prevent some large molecules like some large antibiotics from getting access to the outer membrane. Now, in kind of medical microbiology, LPS is also called endotoxin. Now, the basal structure, the lipid A and the core oligosaccharide, these are highly conserved structures shared by all gram-negative bacteria. And cells of our immune system have receptors that can specifically bind to these conserved structures because it's a kind of an alarm system to tell us, hey, we've been infected by gram-negative bacteria, we've got to do something here, right? So if you were to have the terrible misfortune of being injected with a large amount of LPS into your bloodstream, where you'd have fever, if you get a lot in your gut, you'll have diarrhea, and if you've got a systemic infection with gram-negative bacteria, you could have septic shock. That's all because of, the reaction of your body to the presence of endotoxin, LPS. So that's the importance of LPS for us in infectious biology. And the other important thing to know about LPS is that these repeated sugar motifs up here that are variable between strains of the same species, they're very useful for identification of bacteria. And these are what's called the O antigen. O is for O's, right? So they're sugars. Okay, so how is LPS synthesized? It's the same problem for bacteria. It's a complicated polymer on the outside of the cell. So it's the same type of problem as for the synthesis of peptidoglycan. Now, what's lucky for us is that, it's the same type of problem, so the solution is very similar as well. So what happens is this. So the core oligosaccharide is synthesized in the cytosol, bound onto the lipid A part. Then there's a flipase that's going to flip the whole thing over so that this becomes transported into the periplasm. So that's for the core oligosaccharide lipid A. How about the O antigen repeats? Well, it's just like NAM, NAG, tetrapentapeptide, right? This is synthesized on bactoprenol diphosphate on the cytoplasmic, in the cytosol. Then another flipase is going to turn it over, so you have this repeated sugar motif, 2 phosphates here on bactoprenol, and then a transglycosidase enzyme, which is going to add this subunit onto the end of a growing chain. So it's exactly the same mechanism here. Synthesize the repeated subunits in the cytosol on bactoprenol, flip them over in the periplasm, then a transglycosidase makes the chain longer. Then after a certain number of repeated subunits have been added, the whole thing gets transported onto the core oligosaccharide. So that's it. You've got your LPS molecule here in the periplasm. And the last step is that there is a big multi-protein complex which is going to grab this thing and shift it all the way up to the outer side of the outer membrane. And of course, this costs energy to do that. Okay, so you don't have to know this in detail, but the general idea that, you have something similar, the repeated subunits on bactoprenol, the core antigen synthesized on lipid A, flipped into the periplasm, polymerization with transglycosidase, and then it goes out. So general steps in the process, I think you should know that. Okay, next thing is the capsule. So capsule antigens are facultative. There's a polymer on the, right on the outside of the bacteria. You can see them as this kind of like diffuse layer around the bacterial cell. On electron microscopy, they can look like this a little bit. So most of these polymers are sugars, polysaccharides. Sometimes they can be called a glycocalyx. That's what this means here. Not always, though. Some capsule antigens are polypeptides. And they can be present in gram-positives and gram-negative bacteria. So they are not essential components. Within a species, you can have some strains that have capsule antigens, some strains that don't. And if a capsule antigen is present, it can have different types of sugars in the polymer. Okay? So that means that it's also useful for identification. And this time they're called the K antigens. K for Capsule in German. Now, it's useful for us to be able to identify bacteria, but that's not the function of this structure. That's not why it exists. They exist in order to protect bacteria against, a bit like LPS, detergents, antibiotics, especially for gram-positive bacteria which don't have an outer membrane. Capsule can be important for this. The capsule antigens have a role in protecting bacteria from bacteriophages. Bacteriophages are viruses that infect bacteria. And these viruses need to attach to receptors that are on the surface of the bacteria. Now, if the receptors which are here are covered by a massive capsule antigen, a bacteriophage particle coming in here will not be able to attach to its receptor. So these bacteria will be resistant to phage. And some of these polymers are very efficient at retaining water, so they can prevent bacteria from drying out if they're on a surface, so water retention. And finally, in infection biology, they are important virulence factors because they can prevent neutrophils and macrophages from phagocytosing bacteria and killing them. So Streptococcus pneumoniae, pneumocoque, the pathogenicity of this bacteria is very closely related to whether it's got a capsule antigen or not. Okay, the non-capsulated strains of these bacteria are not invasive and not really pathogenic. Okay, so important virulence factors. Right, so what else can we say? Synthesis again. Same problem, same kind of solution. How to synthesize a polymer outside the cell. And it's really a very similar system. So you've got firstly the repeated subunits. They're synthesized on bactoprenol pyrophosphate. Then there's a kind of enzyme which is going to flip this structure over and transport it to the periplasmic face. And then a transglycosidase which is going to start polymerizing the polymer here. And then once it's big enough, the whole thing gets transported out to the surface of the bacterium. So, similar kind of process, okay? Right, oh yeah, we've got to do these filaments now. Yeah. Okay, so now we're going to take a look at filamentous structures on the surface of bacteria. So first we have firstly fimbriae and pili. So that means all these kind of little spikes sticking out of the surface of this bacterial cell, not the flagella here. Okay, so they're about 10 nanometers or less in diameter, and their function is either to absorb, attach bacteria to surfaces or host cells for pathogenic bacteria. Some of them are hollow, so they're like little needles that can eject stuff. So there's like for secretion, and some of them can be used by bacteria to kind of walk around on a surface. So several different functions for fimbriae and pili. Now you might wonder why we have two names for the same thing. I know it's something that annoys me every single year. And this is because you had two teams of microbiologists describing these structures pretty much at the same time in the 1950s. So one group called them fimbriae. So this is for Latin, from Latin apparently means like threads. And another group called them pili, like hair, right, like poil. Okay, so two groups, each group gives them their own name and nobody wants to like back down. So also kind of similar structure are the conjugation pili. And there are fewer of these per cell, maybe up to 10, but generally 1, 2, or 3. They have a slightly bigger diameter, and these are used for conjugation. That's the exchange of genetic information between bacteria. So during conjugation, one of these bacteria is going to throw out a conjugation pilus, which is going to attach onto the other one. Then it's going to kind of reel it in, and they're going to get very, very close and then exchange DNA. So we have absorption, secretion, gliding motility, and conjugation. We've got like 4 different functions here. And okay, so that's a bit more complicated than LPS, capsule, which is just kind of like big layer outside protecting the bacterium. So the functions are a bit more complicated, and I'm afraid this is associated with different kinds of structures being responsible for these different functions. Now, I'm going to spend a little bit of time trying to explain the important points in this diagram here. Now, what you need to know and understand is which type of pili do what and why. Okay. Now, when you talk about different types of pili, you can see we got type 3, we got type 4. Well, where's type 1 and type 2? Huh? Well, from what I understand, when microbiologists were trying to classify, Fimbriae and pili into different, groups, they started out with a serological classification. So there were groups 1, 2, 3, and 4. And then later on, through molecular biology, when people started to understand a bit more about the structures of these different types of pili and how they were produced, well, it was realized that groups 3 and group 4, that's okay. These classifications make sense, but the previous classification, type 1 and 2, didn't really make any sense, okay? So type 3 exists, type 4 exists, type 1 and 2 don't exist. So what exists now is a classification of CU pili, curly pili, type 3 and type 4. And this is, okay, this classification is relevant to gram-negative bacteria. Okay, you can see this is internal membrane, external membrane. This is true for gram-negative bacteria. Now, gram-positives, they have some pili, a bit like these two types, CU and curly. Something similar to this exists in gram-positives. But they're not really attached to the outer membrane. They're probably attached to the peptidoglycan directly. But they don't have like type 3 and type 4. Okay, so that's why we have different types of classification. Right, so let's just think about CU and curly. So how are these structures produced? They're anchored in the outer membrane, and the protein subunits that make up the filament are synthesized in the cytosol, then they're exported into the periplasm, and then for CU pili, they are added to the base of this structure. So you get more subunits added here, and then the whole thing gets pushed out of the cell. Okay. For curly pili, the protein subunits that make up the filament, they're exported, outside the cell, and then they bind onto the top. And the function of CU and curly pili is just adhesion onto surfaces or onto other types of cells. The filament is not hollow, okay, so nothing can go through the inside of these pili. They can't be used for secretion. And once they've been polymerized, all the polymerization happens in the outer membrane or outside the cell. So once they're polymerized, that's it, they just stay there. Okay, they can't be retracted, the bacteria don't really control what happens afterwards. So they're just involved in adhesion. Okay, type 3 pili. Okay, what happens here is that you've got a big structure of multiple proteins that spans the plasma membrane, peptidoglycan layer, and the outer membrane. And then you have a hollow tube which forms the filament. Now, the filament proteins are synthesized in the cytoplasm, and then they come up through the middle of the tube here, all the way up there, they come out and they polymerize on the top of the tube. And that's the way that the different substrate proteins that are transported can be exported through a type 3 secretion system. So the filament proteins, they go up through the tube, they bind on the top, and then afterwards, the different proteins that are going to be excreted, they're also, sent out through this tube. Now, often, type 3 secretion systems are virulence factors for pathogenic bacteria. And the proteins that are ejected here or exported from the bacterium can be bacterial exotoxins that are injected into the host cell to try and kill them or change their biology. Okay, that's type 3 secretion pili. Now, type 4 pili, also complicated structures with a big structure that spans the plasma membrane, the periplasm, and the outer membrane. But in this case, the filament proteins are added at the base here. So they're added here, and then this will push up the whole filament. So the filament can be extruded. What's very important here is that this process can be reversed. So that depending on signals coming into the bacterial cell, the type 4 pili can be extruded or they can be retracted. And this is very important because this allows the type 4 pili to be used for gliding motility, as we'll see in a minute. And type 4 secretion pili kind of combine these two elements. Okay, so type 4 secretion pili, the filament is a hollow tube, so molecules can be transported inside the tube and secreted. And also, these filaments can be extruded and then retracted. So, conjugation pili, the F pilus used for the transfer of genetic material, they're really a modified form of type 4 secretion pili, okay? Because they're sent out from one bacterium to another, then they're retracted, and then DNA transits through the tube. Okay, so one other thing about this, so adhesion pili, type 4 or Cu or curly, they're also virulence factors for bacteria for pathogenic bacteria, because the first step in the infectious process is for the pathogenic bacterium to bind to the host tissue and invade the host. So sometimes whether strains of bacteria are pathogenic or not depends on whether they can bind effectively to host tissues. So these adhesion molecules are often pili and fimbria. Okay, so type 4 pili and gliding motility, how does this work? So you've got kind of little bacillus there on the diagram, which is attached to a solid surface through its type 4 pili. What is going to happen is they're going to extrude new pili at the front and then retract pili at the back. Then these pili are going to attach to the surface and then they'll be retracted. And this generates force to move the whole thing forward. Okay? So it's kind of like throwing out a rope and then drawing it in and then throwing out a rope again and pulling it in and doing like this. So it moves around like that. Okay? So that's gliding motility. Okay, only possible for type 4 pili. Okay, I'm just going to... Do you want to... Oh, I didn't put the code on there either. Do you want to try and have a go at this one or not? Or is it a bit obvious since I just explained? Okay, have a go on it and I'll try and get the QR code. Okay, stop. Oh, I didn't put the question on here yet. Okay, we'll just have to tell me. Okay, so everybody thought about this. Which one is the correct answer? Who says 1 is the correct answer? Nobody. Who says 2 is the correct answer? Who says 3 is the correct answer? Some people. Who says 4 is the correct answer? Okay, the people who put your hands up for 3, you're right. You're right, that's why. Okay? No. Yeah, so So for type 3 pili, this polymerization is at the tip here. So it can't be controlled. It can't be reversed by the bacterium, okay? Whereas type 4 pili, the subunits are added at the base, and you know that's accessible to control because it's part of this structure that's in contact with the cytoplasm. So it can be controlled. Man. Okay, so that's what we got. Gliding motility. Okay, now flagella. Okay, so flagella is like a kind of whip, right? That's what it means. Now, flagella can be present or absent on bacteria. And if they are present, they can either be, you know, one copy, one flagellum at the end of the cell, in which case they are called monotricious. There could be several flagella at the same end of the cell, in which case they are called lophotricious. Or they can be multiple flagella all around the bacterium, in which case they're called peritricious. So, I suppose you could remember, okay, Pseudomonas called 1, Proteus have got them all over the place. Now, looking at the structure of the flagellum, it's complicated structure with anchored in plasma membrane, kind of basal structure like a kind of axle that traverses the peptidoglycan layer and is anchored also in the outer membrane in a hook structure, and then the filament over here. The filament is like got a diameter of 20 nanometers, 15 to 20 microns long. So the bacterial cell itself is going to be something like 5 micrometers in length. So this filament, you know, it's like 3 or 4 times longer than the whole bacterium. Think about if you're yourself, like with a big kind of chain, like hanging around 4 times your own body length, you kind of swing it around or something. Okay, so there are thousands of copies of the filament protein in the filament here, and this is what constitutes the H antigen, which is used in identification. So H means heat sensitive, okay, because they're made of protein, so they are denatured by heat. Now that's not true for LPS, the O antigens, and the Capsule K antigens, which both resist heat. So H is the heat sensitive antigen filament proteins. Okay, so when bacteria, okay, within a strain, bacteria can either have a filament or not, and or have a flagellum or not, and if a flagellum is present, the filament protein can have a different structure, so you have different flagellar antigens. But, you know, each bacterium will only be expressing one type of flagella, okay? Right, so how does it work? So how does this thing generate movement? Well, what happens is that this whole basal structure here will spin around like the kind of like, I don't know, an axle or moving in a car or something. And the energy to produce this movement comes from the translocation of protons in the periplasm through to the cytoplasm. And this proton motive force generates mechanical force. Now, flagellum means like a whip, right? So you imagine the thing's going to kind of be whipping around like this. That's not the way it works, okay? So the base plate is spinning around and the whole filament is going around like this. And it can spin around in 2 directions, and this is controlled by the switch protein down here. So depending on its status, I don't know if it can control whether the flagellum rotates in a clockwise or an anti-clockwise sense. And that's important to control movement. Well, one last thing, yeah, filament proteins, how do they get synthesized? So the major filament proteins, they're synthesized on the ribosome, and then they're transported up the inside of the basal body through the filament, which is hollow, and they attach onto the distal tip. So does that remind you of anything? What's it like that we've just seen? Yeah. Yes, it's very similar to a type 3 secretion pilus or pili. And so that's telling us something about the origin of flagella. Okay, this is a complicated molecular machine. All the parts work together to produce some complicated function, which is moving the bacterium about. If you have to make this structure from nothing, that's a very difficult problem in evolution. But in fact, flagellated bacteria don't start from nothing. They start with type 3 secretion system, which already exists, and you just got to like attach a motor on the bottom of it and the thing's going to start spinning round. Okay, so it's not so difficult. Now, one of the things I need to say about this is, okay, once again, the example here is from a gram-negative bacterium, but flagella also exist in some gram-positives with a different basal structure, okay? Right, so that's a structure, that's a synthesis, similar to type 3 secretion pili. Now we need to understand how flagellar motion can be used by bacteria to do something intelligent. I don't know if it's the right word, but something that makes sense in biological terms. So the function of flagella is to move bacteria around in a liquid. Okay, so they're swimming around in a pool, in a swimming pool. So that means they've got to navigate in 3 dimensions. Now, if you or I were trying to design something like this, you'd probably say, okay, we need something that moves up and down, and then we need something that decides where we're going left and right, and then we need something that decides whether we're moving backwards and forwards. So you've got like 3 axes in your 3-dimensional space, and you'll have like 3 types of ways to move. Well, the flagellum can't do that, okay? It's only got 2 choices. You can turn like this, or you can turn like that. That's it. And somehow the bacterium has got to move around in 3 dimensions just with 2 speeds, forwards, or spinning this way or spinning that way. So how does that work? Well, when the flagellum is rotating in a counterclockwise sense, the net effect is the bacterium is going to move forward in a straight line. And if it rotates in a clockwise sense, then the bacterium is going to tumble around randomly in 3 dimensions. So for bacteria, you don't have forwards and backwards, left and right, up and down. You've got straight or random. Those are your 2 modes of moving around. Now that works for monotricious bacteria and also works for peritricious bacteria. So for peritricious bacteria, they have many flagella all around the bacterial cell, and their rotation will be coordinated. So all the flagella on the bacterium will be rotating in the same sense, okay? So in a peritricious bacterium, all the flagella rotate either in a counterclockwise sense or they all rotate in a clockwise sense. If they're all rotating in a counterclockwise sense, it's going to move forwards. If they're all rotating in a clockwise sense, the whole thing is just going to tumble around randomly. So 2 modes of locomotion, forwards and random, and alternating between these 2 modes is what allows the bacteria to move towards interesting stuff, like glucose, amino acids, and can move away from things they don't like, I don't know, UV radiation, something like that, or high acid, low pH conditions, something like this. So how does that work? Okay, I'm going to hope you appreciate my wonderful slide here. It took me such a long time to create, because before I would try and draw this out, you see, but I thought, okay, we need to try and be able to record everything this year, so I made this wonderful diagram, which I hope you can appreciate. So here we go. We've got a source of glucose here, some kind of solid material in a liquid, and the glucose is dissolving and it's creating a concentration gradient. So nearby, this is high concentration of glucose. Far away, it's very low concentration. Now, the bacterium, it's going to be useful for the bacterium if it can move towards the glucose, right? Because there's a lot of stuff to eat over there. So it's going to apply a very, very simple algorithm. So starting out, you've got clockwise flagellar rotation, and that's going to last for kind of random duration. So for a while, this bacterium is going to kind of spin around randomly until after a while it stops and the flagellar rotation changes orientation. So it's going to start moving in a straight line because the flagellum is going to be turning now in a counterclockwise sense, and then it's going to start moving in a straight line. And as long as the bacterium is moving up the concentration gradient, that is getting closer to the glucose, the flagellar rotor is going to carry on spinning around in the same sense. Now at some point, the bacterium is going to overshoot and it's going to start getting further away from the glucose. And when that happens, where it's going to the flagellar rotor is going to flip it's going to change, it's going to start turning the other way, and then it's going to start randomly changing direction. And then it will start moving in a straight line again. Okay? So you could probably write out this algorithm in 10 lines of Python or something, and it would work, all right? You could, set a robot to do this, and it would get closer to the center of whatever it is you're trying to detect. And that's how, gram-negative bacteria with flagella can actually move towards something that is going to be good for them. They have a simple algorithm that works with biochemistry inside the cell. Okay, so, if you want, well, you could see what's going to happen. You're going to get further away after a while, then it's going to randomly change direction. If it randomly changes direction and moves away, then it won't move very far, okay? The time that it moves away is going to be very short, then it will change direction again. And as long as it's getting closer, it's going to have a tendency to keep moving in a straight line. So what's going to happen is that, from here, it's going to move over there and then around here, and then it's going to carry on moving, but within a region that's really quite close to the peak of the concentration. So, yeah, that's how flagellar motility is used by bacteria for chemotaxis. Okay, yeah, now you can answer this question if I can go and open it up. Okay, so which structures are used for identification? So that should be started. Okay, good. Okay, 39 votes. I guess some people voted more than once. Oh, maybe it means there's more than, yeah, because you can choose more than one thing, right? Yeah, so that's one. Okay, yeah, so basically, yes, you're right. Okay, LPSO repeats, correct. Capsule antigens, correct. Peptidoglycan, no, it's not useful because it's highly conserved. All gram-positives, all gram-negatives have it. It's not going to help, okay? It's the same, pretty much the same for every type of bacterium. That's the same kind of problem for the LPS core oligosaccharide. It's highly conserved, certainly by different species within the same genus. So it's not really going to be very useful. CU pili, that's almost like the opposite problem. It's too variable, okay? You don't just have one type per bacterial species or strain of bacteria. So just with one isolated bacterium, you'll have like you might have 3 or 4 different types of CU pili on there, or 1 or 0. So it's almost like too variable to be useful. Okay, so these are the ones. When they're present on the bacterium, you just have one type, and they're sufficiently variable to be useful. Okay, so that's that. Right, where are we now? Okay, so we've got about 10 or 15 minutes, or until I faint from heat exhaustion, whichever comes first. So that's it. We've finally finished the external structures of the bacterial cell, and now we're going to move on to the internal structures. Unfortunately, it's quite a lot simpler, okay? So one of the things you can find is inclusion bodies, which are storage structures for carrying nutrients. So they can be organic or inorganic. So, in terms of organic inclusion bodies, they're constituted of polymers of glycogen, polyhydroxybutyrate, so these are going to be storage of carbon, right? Or cyanophycin, which is nice because there you've got a polymer of arginine and aspartate, so you can store nitrogen and carbon. Now, inorganic inclusion bodies can be polyphosphate, so phosphorus, it's essential for bacterial growth, and it can often be the limiting factor. So if bacteria are growing in conditions of abundant phosphorus, then they can take up more than they need and store it in a polyphosphate granule, because a bit later on they might find out that all the phosphate's gone and they'll still have a stock to carry on growing, Okay, some bacteria base their energy metabolism on sulfur oxidation. So similarly, this can be a limiting chemical resource for growth, in which case, they can store granules of sulfur inside the cell. Now, these inclusion bodies can just be directly in contact with the cytoplasm, which is true for glycogen and polyphosphate and cyanophycin granules, or they can have a kind of like lipid or protein shell around the inclusion body. Now, that's true for beta-hydroxybutyrate and sulfur. So that's all I really need to say about that. Okay, so the nucleoid, so this is the region of the cytoplasm of the bacterium that contains DNA. Now, inside a bacterium, the DNA isn't just a big massive molecule of deoxyribonucleic acid floating around in solution. Because if that were to be the case, it would be far too big to fit inside the cell. What you can do is digest a bacterium, degrade all the proteins with proteases, and this will liberate the bacterial chromosome as free DNA. So you can see the DNA genome, if it's allowed just to be like free, like this is much bigger than, takes up much more space than you have available inside the cell. So for this to fit into the cell, it's got to be compacted. And that occurs not through histones, like in our nuclei, but by complexing with polyamines, like spermine, spermidine. So you've got the negative charge on the DNA, it's neutralized by the polyamines, positive charge. And also something called HU proteins are in many bacteria. which introduce bends in this compacted DNA. So, you know, the DNA can loop around in a compacted form, and that's what the nucleoid is composed of. DNA, polyamines, and other bacterial proteins to keep it all compacted. And at one point, the nucleoid is going to be attached to the cell wall. Now, there are exceptions. So in archaea, some archaea have got histones. And another weird exception is in some planktomycetes, you have this kind of arrangement, which shouldn't really exist in bacteria, because you've got the DNA in the nucleoid, it's in a membrane-bound compartment. Now that is really weird, okay? This looks like a bacterium with a nucleus. And you've got something around here that looks like a nuclear pore. So this is very, very rare and exceptional, but it kind of shows you that there's some weird stuff in the world of bacteria. Yeah, I've got 10 minutes, so I'll go through the cytoskeleton and then we can all have a break, okay? Okay, so the bacterial cytoskeleton. Classically, one would say that the cytoskeleton is specifically eukaryotic and that bacteria do not have an organized cytoskeleton. So that's generally true, but in fact, when you look closely, there are bacterial homologs of actin and tubulin proteins. And the bacterial homolog, which structurally is similar to actin, are the MREB or MREB-like MBL proteins. So like actin, they can exist as a soluble or polymerized form. And when they're polymerized, they form these kind of spiral polymers inside bacilli. So these are what forms a structure that allows the bacterial cell to elongate. So they are only present in bacilli, not in cocci, okay? And if you knock out MRE proteins in MREB proteins in Escherichia coli, for example, instead of having a kind of a coccobacillus form, they become round, okay, spherical. So, you know, whether you've got cocci or bacilli in terms of morphology depends on whether the bacteria have got MREB and MBL proteins. That's their function, elongation of the bacterial cell. Now, the bacterial homolog of tubulin is a protein called FTSZ. And similarly, it can be found in bacteria as a soluble monomer or as a polymer. And it's very interesting because its function is to help organize the separation of the bacterial chromosomes and the division of the bacterial cell during growth. So what happens is that during cell division, the monomers of FTSZ will form a ring around the center of this bacterial cell that's growing, and the two copies of the bacterial chromosome that are getting replicated will migrate to opposite ends of the bacterium. And then once they separate, this ring of FTSZ will control the accumulation of proteins that are going to divide the bacterium into two. So FTSZ has got a very, very fundamental role in bacterial cell division. Now, that is really, well, I think I'm kind of amazed about this because, okay, gamma tubulin in eukaryotic cells, its essential function is to form the structures that, well, okay, I don't have a pen here. So think about mitosis, right? During mitosis, you've got two mTOC. You know what that means? Mitotic something organizing center. Okay, you've got two centrioles like this, and you've got the mitotic spindle, which is formed with tubulin fibers. Okay, so gamma tubulin is the tubulin that forms these centrioles here, that are the base for the formation of the mitotic spindle. It's an essential function for chromosome segregation in eukaryotic cells. FTSZ, its closest homologue is gamma tubulin. All right, so there are massive differences in the mechanism of chromosome segregation in bacteria and eukaryotic cells. The whole system is very, very different. But at the base of it, you've got these two proteins, FTSZ and gamma tubulin, that are very similar in structure. So it makes you think that maybe, way back in the beginning of, the evolution of eukaryotic cells, maybe the mechanism of chromosome segregation started out from something a lot simpler with a bit more homology to what happens in bacteria. Okay, so I think that's probably a good point for me to have a break. So we can all go away for 10 minutes. No more than 15 anyway, and I'll see you in a little while.